In December of 2018, a team of American scientists drilled through more than 1,000 meters of Antarctic ice, breached the ceiling of a hidden subglacial lake, and pulled water samples to the surface that contained something no one had expected to find. Not just living microbes, though those were present in abundance. Not just the chemical signatures of an active ecosystem, though those were clear. What the team found, preserved in the sediments at the bottom of this dark, sealed body of water, were the remains of organisms that had lived and died in the region long before the ice sheet existed. Tiny crustacean shells, fragments of microscopic animals, wisps of organic material that bore the unmistakable signatures of a biological world that had once thrived in open water, under open sky, in an Antarctica that looked nothing like the frozen continent we know today. The lake was called Mercer, subglacial Lake Mercer to be precise, named after the American glaciologist John Mercer, who in 1978 published one of the first warnings that the West Antarctic ice sheet might be vulnerable to collapse under the influence of rising global temperatures. The lake sits beneath the Mercer ice stream, a fast-flowing tributary of ice that drains the interior of the West Antarctic ice sheet toward the Ronne Ice Shelf. The ice above it is approximately 1,084 meters thick. The lake itself is shallow, a thin lens of water pooling in a depression in the bedrock, connected to the broader subglacial drainage system of the region through channels that carry water downstream toward the coast. The mission to access Lake Mercer was called SALSA, an acronym for Subglacial Antarctic Lake Scientific Access. It was the successor to the Wizard project, which had successfully accessed the nearby subglacial Lake Whillans in 2013, confirming for the first time that living microbial communities existed beneath the Antarctic ice sheet. The SALSA team built on the technology, the protocols, and the hard-won experience of Wizard, deploying an improved hot water drilling system with contamination controls designed to meet the most rigorous standards in the field. But it was the sediment cores, not the water samples, that produced the most startling results. Because buried in the layers of mud at the bottom of Lake Mercer, sealed beneath more than a kilometer of ice, in an environment that had not seen sunlight in thousands of years, were the bodies of creatures that had no business being there. Crustaceans, tardigrades, diatoms, fragments of plants, the remains of an ecosystem that had existed in this exact location during a time when the ice was gone, when the land was exposed, when water flowed freely, under an Antarctic sky that was warmer than anyone in the modern era has ever experienced. The discovery forced a fundamental reconsideration of what we thought we knew about the history of the West Antarctic ice sheet. The conventional understanding held that the ice sheet in this region had been continuously present for at least hundreds of thousands of years, possibly longer. The biological remains in the Lake Mercer sediments suggested otherwise. They suggested that the ice had retreated from this location within a geologically recent time frame, perhaps within the last 120,000 years, possibly more recently, allowing open water and terrestrial ecosystems to exist where there is now only ice and darkness. And if the ice had retreated from this location before, under natural climate forcing, the implications for its future stability under anthropogenic warming were deeply unsettling. The discovery at Lake Mercer was therefore not merely a biological finding. It was a climate finding, a geological finding, and a warning. It told us that the ice we see today, the ice we depend upon to remain in place as a bulwark against catastrophic sea level rise, has not always been there. It told us that the boundary between ice and open water in this part of Antarctica is not as fixed as we had assumed. And it told us that the organisms whose remains were preserved in the sediments had lived and died in a world that the current inhabitants of the planet might one day see again if the ice continues to retreat. The sediments held more than just bodies. They held a timeline, layer upon layer of material, deposited over millennia, recording the history of the environment above. Some layers contained mineral grains consistent with glacial erosion, deposited by ice flowing over rock. Others contained organic material, the remains of biological productivity in open water. The alternation between these two types of deposit told a story of a landscape that had oscillated between ice-covered and ice-free conditions, a landscape that was not permanently frozen, but intermittently exposed, a place where the ice sheet advanced and retreated in response to forces that are still not fully understood. The implications of this oscillation are staggering. The West Antarctic ice sheet contains enough ice to raise global sea levels by approximately 3.3 meters if it were to melt entirely. Current climate projections suggest that the ice sheet is already losing mass at an accelerating rate, contributing to sea level rise that threatens coastal communities worldwide. The question of how quickly the ice sheet can retreat, and whether it has retreated rapidly in the past, is one of the most consequential questions in all of climate science. The sediments at the bottom of Lake Mercer may contain the answer. By the end of tonight, you're going to understand what the SALSA team found at the bottom of Lake Mercer, and why those findings sent shockwaves through the Antarctic science community. You are going to understand how crustacean shells and tardigrade fragments ended up beneath a kilometre of ice, and what their presence reveals about the recent geological history of the West Antarctic ice sheet. You are going to understand the connection between ancient life in a subglacial lake and the modern crisis of ice sheet instability, and why the sediments of Lake Mercer may contain the most important climate record ever recovered from beneath the Antarctic ice. Before we get started, if you find these mysteries as fascinating as we do, take a second to like the video or subscribe. It's a simple action, but it helps this channel reach more curious minds like yours. Now, let's begin. The West Antarctic ice sheet is one of the most closely watched and least understood features of the Earth's climate system. It is a massive body of ice, covering an area of roughly 1.8 million square kilometers, with an average thickness of approximately 2,200 meters. It contains enough frozen water to raise global sea levels by approximately 3.3 meters, a number that sounds modest until you consider what 3.3 meters of sea level rise would mean for the world's coastal cities, island nations, and low-lying agricultural regions. 3.3 meters would inundate large portions of Bangladesh, the Netherlands, and southern Florida. It would displace hundreds of millions of people. It would redraw the coastlines of every continent. The West Antarctic ice sheet differs from its much larger neighbour, the East Antarctic ice sheet, in a fundamental and ominous way. Much of the West Antarctic ice sheet rests on bedrock that lies below sea level, a configuration known as a marine-based ice sheet. The base of the ice sheet in many areas is not perched on high ground. It is sitting in a basin, a depression in the bedrock that was carved by tectonic forces and glacial erosion long before the current ice sheet formed. The bedrock slopes downward from the coast toward the interior, meaning that the ice at the edges of the sheet is perched on relatively high ground, while the ice in the interior rests on bedrock that descends to depths of more than 2,000 meters below sea level. This geometry creates a condition that glaciologists call marine ice sheet instability. When warm ocean water penetrates beneath the floating ice shelves at the coast and melts the underside of the ice, the grounding line, the point where the ice lifts off the bedrock and begins to float, retreats inland. Because the bedrock slopes downward toward the interior, the retreating grounding line encounters progressively deeper bedrock, which means that the ice at the new grounding line is thicker, which means that it flows faster into the ocean, which means that the grounding line retreats even further. The process is self-reinforcing. Once retreat begins on a downward-sloping bed, it tends to accelerate until it reaches a stabilizing feature, a ridge or rise in the bedrock that reverses the downward slope and provides a new anchor for the ice. The Thwaites Glacier, one of the largest and fastest-moving glaciers in West Antarctica, is currently undergoing exactly this kind of retreat. The grounding line of Thwaites has been retreating inland at rates of up to 1 kilometer per year, and the bedrock beneath the glacier slopes steeply downward toward the interior, providing no obvious stabilizing feature for hundreds of kilometers. The potential collapse of the Thwaites Glacier and the broader destabilization of the West Antarctic ice sheet that could follow is one of the most alarming scenarios in contemporary climate science. It has earned Thwaites the informal nickname, the Doomsday Glacier, a label that is melodramatic but not entirely unjustified. Lake Mercer sits within this broader context of West Antarctic instability. The Mercer ice stream, beneath which the lake lies, is one of several ice streams that drain the interior of the ice sheet toward the Ross Sea. Like the nearby Wilkes ice stream, the Mercer ice stream flows at rates of hundreds of meters per year, carrying ice from the continental interior toward the coast. The ice above Lake Mercer, approximately 1,084 meters thick, is part of this flowing system, moving over the lake like a slow-motion conveyor belt, carrying ice from upstream to downstream. The lake itself was identified through satellite observations that detected subtle changes in the elevation of the ice surface above it. Between 2006 and 2007, the ice surface above Lake Mercer rose by approximately half a meter, indicating that the lake was filling with subglacial water from upstream sources. Subsequent satellite monitoring tracked additional filling and draining events, confirming that Lake Mercer is a dynamic feature of the subglacial hydrological system, periodically filling with water that flows through channels at the base of the ice sheet, and then releasing that water downstream when the lake reaches a critical pressure threshold. This dynamic behavior made Lake Mercer an attractive scientific target for several reasons. First, the periodic filling and draining of the lake meant that it was hydrologically connected to the broader subglacial drainage system, potentially harboring microbial communities that were representative of the wider subglacial environment rather than confined to a single isolated basin. Second, the sediments at the bottom of the lake, deposited by flowing water and settling particles over time, could contain a record of past conditions in the subglacial environment, providing a window into the history of the ice sheet and the landscape beneath it. Third, the location of the lake within the Mercer ice stream catchment placed it in a region of West Antarctica that is scientifically important for understanding ice sheet dynamics and stability. The SALSA project was funded by the United States National Science Foundation and involved a large, multidisciplinary team of researchers from universities and research institutions across the country. The principal investigators included John Priscu of Montana State University, who had also led the biological component of the WISARD project, and Sławek Tolaczyk of the University of California, Santa Cruz, who had contributed to the glaciological research at Lake Wilkes. The team also included geochemists, sedimentologists, oceanographers, and engineers, each bringing specialized expertise to a mission whose scientific goals spanned multiple disciplines. The planning for SALSA began years before the drilling campaign, with site surveys, environmental impact assessments, equipment design, and logistical coordination consuming the better part of a decade. The drilling system was an evolved version of the hot water drill used by WISARD at Lake Wilkes, incorporating improvements to the filtration, sterilization, and monitoring systems based on the experience gained during the Wilkes campaign. The contamination control protocols were among the most rigorous ever applied to a subglacial drilling operation, reflecting the heightened awareness of contamination issues that the WISARD success and the Lake Vostok controversy had instilled in the subglacial science community. The logistics of the SALSA campaign were formidable. The drill site on the Mercer ice stream was accessible only by ski-equipped aircraft from McMurdo Station, the primary American research base in Antarctica. All equipment, fuel, supplies, and personnel had to be flown to the site and cached before the drilling season began. The camp on the ice surface was temporary, designed to support the drilling team for the duration of the campaign, and then be removed entirely, leaving no permanent infrastructure behind. The total weight of equipment and supplies transported to the site exceeded several 100,000 kilograms, a logistical effort comparable in scale to a small military operation. The team arrived at the Mercer ice stream in late November of 2018 as the austral summer began, and temperatures on the ice surface rose from their winter extremes to a relatively balmy -20 to -25 degrees Celsius. The assembly of the drilling system, the testing of the contamination control equipment, and the final preparations for drilling occupied several weeks. By late December, the system was ready and the drilling began. The hot water drill melted through the ice at a rate that reflected both the efficiency of the system and the demands of the depth. 1,084 meters of ice, roughly 300 meters deeper than the ice above Lake Williams, required a correspondingly longer drilling period, greater volumes of heated, sterilized water, and more careful management of the borehole to prevent refreezing during the operation. The drilling continued through the Antarctic summer days, the team working in shifts around the clock, monitoring the contamination instruments, adjusting the drilling parameters, and waiting for the moment when the drill would break through the final meters of ice and enter the lake. The breakthrough came on December 26, 2018. The drill nozzle penetrated the ice water boundary, and pressurized lake water surged into the borehole. The contamination monitors confirmed that the water entering the borehole had microbial concentrations significantly higher than the sterilized drilling water, indicating that the organisms in the lake water were indigenous rather than introduced. The sample collection instruments, pre-positioned in the borehole, captured the first clean water and sediment samples from a second subglacial lake in the Williams-Mercer region. The initial excitement of the breakthrough was tempered by the awareness that the real discoveries would come later. In the laboratories, when the samples were analyzed with the full array of modern analytical techniques, the water samples would reveal the composition and metabolic activity of the lake's microbial community. The sediment cores would reveal the geological and biological history of the site, and together they would tell a story that no one on the drilling platform, standing on the ice in the Antarctic summer sun, could have fully anticipated. But what scientists discovered next was worse than anyone expected. Not worse in the sense of a disaster. Worse in the sense that the implications were more far-reaching, more troubling, and more consequential for our understanding of the Antarctic ice sheet than anyone had imagined when the drilling began. To fully appreciate what was found in the Lake Mercer sediments, it is necessary to understand the history of ice in this part of Antarctica and the scientific debate that preceded the Salsa discovery. The question of whether the West Antarctic ice sheet has collapsed in the geological past is one of the most contentious issues in glaciology. The ice sheet exists today because the temperatures in the region are cold enough to sustain it, because the ocean waters surrounding it are cold enough to preserve the ice shelves that buttress its margins, and because the snowfall that accumulates on its surface each year roughly balances the ice that is lost through melting and calving at the coast. If any of these conditions changes sufficiently, if temperatures rise, if ocean waters warm, if snowfall decreases, the ice sheet could begin to lose mass faster than it accumulates, leading to thinning, retreat, and potentially collapse. The geological record provides evidence that the West Antarctic ice sheet has been smaller in the past than it is today. Marine sediment cores recovered from the seafloor around Antarctica contain layers of material that were deposited during periods when the ice sheet was reduced, allowing more sediment to reach the deep ocean. Geochemical analyses of these sediments indicate changes in water temperature, ice volume, and biological productivity that are consistent with significant reductions in the extent of the West Antarctic ice sheet at various times in the geological past. The most recent period during which the ice sheet may have been significantly smaller is the last interglacial, a warm period that occurred approximately 115,000 to 130,000 years ago, when global temperatures were roughly 1 to 2 degrees Celsius warmer than pre-industrial levels. During the last interglacial, global sea levels were approximately 6 to 9 meters higher than today, a level that implies a significant contribution from the melting of polar ice sheets. The Greenland ice sheet is estimated to have contributed roughly 2 to 3 meters to last interglacial sea level rise, leaving 3 to 6 meters unaccounted for. The most likely source of this additional sea level rise is the West Antarctic ice sheet, which contains enough ice to raise sea levels by approximately 3.3 meters if it were to collapse entirely. If the West Antarctic ice sheet did collapse during the last interglacial, the implications for its future stability are profound. The current warming trajectory, driven by anthropogenic greenhouse gas emissions, is projected to raise global temperatures by 2 to 4 degrees Celsius above pre-industrial levels. By the end of this century, well above the 1 to 2 degree threshold that may have been sufficient to destabilize the ice sheet during the last interglacial. If the ice sheet collapsed under the modest warming of the last interglacial, it is reasonable to ask whether it will collapse again under the more aggressive warming of the coming decades and centuries. This question has been debated in the scientific literature for decades, with different lines of evidence pointing in different directions. Some studies of marine sediments and ice cores suggest that the West Antarctic ice sheet has been continuously present for at least the past 400,000 years, surviving multiple warm interglacial periods without major collapse. Other studies suggest that the ice sheet has been more dynamic, retreating significantly during warm periods and advancing during cold ones. The uncertainty arises in part from the indirect nature of the evidence. Marine sediment cores and ice cores record conditions at the ocean floor or the ice surface, not at the base of the ice sheet itself. Interpreting these records requires models and assumptions that may introduce errors. The Lake Mercer sediments offered a fundamentally different type of evidence. Instead of reconstructing ice sheet history from distant marine sediments or from the chemistry of ice cores, the SALSA team had recovered material from directly beneath the ice, from the very interface between the ice sheet and the bedrock below. The sediments at the bottom of Lake Mercer had been deposited by processes operating at the base of the ice sheet, and they contained a record of the conditions that had prevailed at that location over time. If the ice sheet had retreated from this location in the past, allowing open water or terrestrial environments to exist, the sediments would contain evidence of that retreat in the form of biological remains, organic carbon, and other indicators of ice-free conditions. The challenge of interpreting subglacial sediments is considerable. The sediments are deposited by multiple processes, including the settling of particles from the lake water, the erosion and transport of material by subglacial water flow, and the slow deformation of sediments under the weight and movement of the overlying ice. Distinguishing between these processes and attributing specific sediment layers to specific environmental conditions requires careful analysis of the grain size, mineralogy, chemical composition, and biological content of each layer. The presence of biological remains in the sediments is particularly diagnostic, because certain types of organisms can only live in specific environmental conditions. Diatoms, for example, are photosynthetic algae that require light. Their presence in subglacial sediments implies that the sediments were deposited during a period when the environment received sunlight, meaning that the ice was absent. The SALSA team recovered multiple sediment cores from the bottom of Lake Mercer using gravity corers and piston corers lowered through the borehole into the lake. The cores were carefully sealed and transported to laboratories in the United States for analysis. The initial descriptions of the cores revealed a complex stratigraphy, a sequence of layers with varying textures, colors, and compositions that told a story of changing conditions over time. The scientific team that analyzed the sediment cores included specialists in sedimentology, micropaleontology, geochemistry, and geochronology, each bringing a different set of tools and perspectives to the interpretation of the material. The analysis was painstaking, requiring months of laboratory work to characterize the physical and chemical properties of each layer, identify and catalogue the biological remains, and develop a chronological framework for the sequence of events recorded in the sediments. The ice core record from other Antarctic drilling sites provided important context for the Lake Mercer findings. The Epica Dome Sea ice core, drilled at Concordia Station in East Antarctica, provided a continuous climate record extending back approximately 800,000 years, documenting the temperature variations associated with 8 glacial interglacial cycles. The Vostok ice core, drilled at the Russian research station of the same name, provided a complementary record spanning approximately 420,000 years. These ice core records showed that global temperatures during the last interglacial were indeed warmer than pre-industrial conditions, consistent with the hypothesis that the warming was sufficient to affect the stability of the West Antarctic ice sheet. The ocean sediment record from the Ross Sea, the body of water into which the Mercer ice stream ultimately discharges its ice, provided additional context. Cores recovered from the Ross seafloor by previous drilling projects, including the Andrill program that operated from the McMurdo Ice Shelf in 2006 and 2007, had revealed cycles of ice advance and retreat in the Ross Sea region over the past several million years. These cycles, driven by variations in Earth's orbital parameters and amplified by feedbacks in the climate system, had periodically brought the edge of the ice sheet far inland, exposing the continental shelf and Ross Sea floor to open ocean conditions. The Andrill results suggested that the Ross Ice Shelf, which currently buttresses the ice streams draining the West Antarctic ice sheet, had collapsed and reformed multiple times in the geological past. The convergence of these independent lines of evidence, from marine sediments, from ice cores, from geological surveys, and now from the subglacial sediments of Lake Mercer, created a picture of a West Antarctic ice sheet that was far more dynamic, far more responsive to climate forcing, and far more vulnerable to collapse than the stable, enduring feature that popular imagination often depicts. The ice sheet is not a permanent fixture of the Antarctic landscape. It is a feature of the current climate regime, maintained by a specific set of temperature, ocean, and atmospheric conditions that have prevailed during the current glacial period. When those conditions change as they have changed in the past and are changing again now, the ice sheet responds, and the biological remains in the Lake Mercer sediments provided the most direct evidence yet that the ice sheet's response can include wholesale retreat from regions that are currently buried beneath a kilometre of ice. The scale of the SALSA operation itself tells a story about what is required to do science at the boundary of the known world. The total cost of the SALSA project, from initial planning through field operations and laboratory analysis, exceeded $10 million, funded primarily by the United States National Science Foundation through multiple grants to the participating institutions. The field campaign alone involved transporting approximately 500,000 kilograms of equipment and supplies to a remote site on the Mercer ice stream, a location with no permanent infrastructure, no roads, no buildings, and no power supply. The drilling system required approximately 400,000 litres of water to be melted from surface snow, filtered, sterilised, heated, and pumped through the borehole during the drilling operation. The water treatment system filtered the drilling water through progressively finer stages, removing particles and organisms down to submicron sizes, and then irradiated it with ultraviolet light to inactivate any microbes that survived the filtration. The entire process was monitored in real time by instruments that measured the biological cleanliness of the drilling water at multiple points in the system. To put the scale in perspective, the drilling camp on the Mercer ice stream was roughly the size of a small village, with heated tents for sleeping, cooking, laboratory work, and equipment storage, a drill tower and associated machinery, fuel storage tanks, generators, and a groomed ski runway for aircraft operations. The camp housed a team of approximately 50 people during the drilling period, all of whom had to be fed, housed, and supported in an environment where the temperature rarely rose above -15 degrees Celsius, and frequently dropped below -30, where winds could reach gale force with little warning, and where the nearest medical facility was a multi-hour flight away at McMurdo Station. The human effort involved in the SALSA campaign was immense. The team included drillers who operated the hot water system around the clock in shifts, engineers who maintained the equipment and troubleshot technical problems, laboratory technicians who processed samples in field conditions, cooks who prepared meals for the crew, and support staff who managed logistics, communications, and safety. Many team members had spent years preparing for the campaign, developing equipment, testing protocols, and conducting preliminary field work in Antarctica during earlier seasons. The SALSA drilling campaign was not a single season of effort. It was the culmination of a multi-year program that began with the earliest planning meetings and site surveys years before the drill was deployed. The depth of the drilling, 1,084 metres, was roughly 300 metres deeper than the Wissard drilling at Lake Williams, which had penetrated approximately 800 meters of ice. This additional depth was not merely a quantitative difference. It introduced qualitative changes in the drilling process. The longer borehole took more time to complete, increasing the risk of refreezing. The greater depth required more water to be pumped through the system, increasing the logistical burden on the water treatment plant. The higher pressures at the bottom of the borehole affected the behavior of the drilling water and the dynamics of the ice-water interface, and the additional 300 meters of ice represented 300 more meters of potential contamination pathway, 300 more meters through which surface organisms could potentially be transported into the subglacial environment. The contamination control challenge at Lake Mercer was therefore even more demanding than at Lake Williams. The SALSA team responded by implementing improvements to the Wissard contamination control system based on lessons learned during the earlier campaign. The filtration stages were upgraded, the UV sterilization capacity was increased, the monitoring instruments were refined to provide more sensitive and more reliable measurements of biological contamination in the drilling water, and the sample collection protocols were tightened, with additional controls and blank samples introduced to strengthen the chain of evidence linking the organisms detected in the lake to the subglacial environment rather than to the drilling process. The investment of resources, talent, and institutional commitment that the SALSA project represents is a measure of the scientific community's conviction that the subglacial environment contains discoveries of extraordinary importance. No nation spends tens of millions of dollars to drill through a kilometer of ice in one of the most hostile environments on Earth unless the questions being asked are of the highest significance. And the questions that SALSA was designed to answer about the biology of the subglacial environment, the history of the West Antarctic ice sheet, and the connection between the two were indeed among the most consequential questions in contemporary Earth science. The answers that emerged from the Lake Mercer sediments would justify that investment many times over in ways that the team could not fully anticipate when the drill first broke through the ice on December 26, 2018. The sediments contained a record that was richer, more complex, and more troubling than anyone had expected, a record that would force the scientific community to reconsider fundamental assumptions about the stability of the Antarctic ice sheet and the vulnerability of the frozen continent to the warming that human activities are driving. And this is where the answer becomes deeply unsettling. The scientific framework for understanding the subglacial environment beneath the West Antarctic ice sheet had been evolving rapidly in the years leading up to the SALSA campaign. The Wissard discovery of living microbes in Lake Williams in 2013 had demonstrated that life existed beneath the ice, but it had also raised a host of new questions about the nature, extent, and history of the subglacial biosphere. Were the organisms found at Lake Williams representative of the broader subglacial environment? Did the microbial community vary from site to site, reflecting local differences in geology, chemistry, and hydrology? And what could the subglacial biosphere tell us about the history of the environment in which it existed? This last question was particularly intriguing because it linked biology to geology in a direct and powerful way. Organisms leave traces of their existence in the environments they inhabit. Their cells, their metabolic products, their shells, their DNA. All of these become incorporated into the sediments and waters of their environment, creating a biological record that can be read by scientists trained in the interpretation of such materials. If the subglacial environment beneath the West Antarctic ice sheet had ever been ice-free, if the ice sheet had retreated and allowed open water or terrestrial ecosystems to exist in what is now a dark, frozen, pressurized environment, then the sediments at the base of the ice sheet should contain evidence of those past ecosystems. The search for this evidence was one of the primary scientific objectives of the SALSA mission. The team designed its sampling strategy to maximize the recovery of sediment cores from the bottom of Lake Mercer using instruments that could penetrate the lake floor and extract cylindrical sections of sediment with their internal stratigraphy intact. These cores, each a few centimeters in diameter and up to several tens of centimeters in length, represented cross-sections through the geological history of the site, with the youngest sediments at the top and the oldest at the bottom. Reading these cores, layer by layer, was like reading a book written by the environment itself, each page deposited by a different combination of processes, operating at a different time under different conditions. The analytical techniques available for reading this book had advanced enormously in the decades since the first subglacial lakes were discovered. High-resolution microscopy could identify individual microfossils, the tiny shells and skeletal remains of organisms in the sediment layers. DNA extraction and sequencing could detect the genetic material of organisms that had lived and died in the environment, even if their physical remains had decomposed. Radiocarbon dating could determine the age of organic material in the sediments, placing specific layers in a chronological framework, and geochemical analyses could characterize the environmental conditions under which each layer was deposited, identifying signatures of marine, freshwater, glacial and terrestrial environments. Together, these techniques gave the SALSA team the tools to reconstruct the history of the Lake Mercer site with a level of detail that would have been impossible even a decade earlier. The question was whether the sediments would cooperate, whether they would contain sufficient biological and geological evidence to tell a coherent story about the past conditions at the site. The answer, as the initial laboratory analyses would soon reveal, was yes. The sediments cooperated beyond anyone's expectations. The sediment cores from the bottom of Lake Mercer told a story that the scientific community was not prepared to hear. Under the microscopes of laboratories in Montana, California, New York, and half a dozen other institutions, the layers of mud and silt extracted from beneath 1,084 meters of ice yielded their secrets. And those secrets were not comforting. The first surprise was the biological remains. The sediment cores contained the exoskeletons of tiny crustaceans, the shells of diatoms, fragments of what appeared to be tardigrades, and traces of plant material. These were not the kinds of organisms that could live in the current subglacial environment. Crustaceans need open water and, in most cases, some connection to the surface ecosystem for their food supply. Diatoms are photosynthetic algae that require light to survive. Tardigrades, the famously resilient microscopic animals capable of withstanding extreme desiccation, radiation and temperature, are nonetheless surface-dwelling organisms that depend on environments very different from the dark, pressurized, nutrient-poor world beneath the Antarctic ice sheet. And plant fragments, by definition, come from environments where sunlight, air, and soil are available. The presence of these organisms in sediments buried beneath a kilometer of ice meant one of two things. Either the remains had been transported to the lake bottom from somewhere else, carried by subglacial water flow from a location where these organisms currently live or recently lived, or they had been deposited at this location during a time when the environment was radically different from what it is today. The first explanation seemed unlikely. The nearest location where crustaceans, diatoms, and plants could live in the open is hundreds of kilometers away, at the edges of the Antarctic continent or on offshore islands. The subglacial drainage system, while active, does not transport material over such distances in ways that would preserve delicate biological structures like crustacean exoskeletons and diatom shells. The second explanation was more troubling and more scientifically consequential. If these organisms had lived at the site of Lake Mercer itself, the ice must have been absent, the landscape must have been exposed, open water must have existed, receiving enough sunlight to support diatoms and enough warmth and nutrients to support crustaceans and other small animals. The area that is now buried beneath more than a kilometer of ice must have been, at some point in the geologically recent past, an open water environment resembling a shallow lake, pond, or coastal lagoon, complete with photosynthetic organisms, grazing animals, and probably an entire food web extending from primary producers to consumers. The initial dating of the organic material in the sediments using radiocarbon analysis produced results that were difficult to interpret. Some of the organic carbon in the sediments was too old to be dated by radiocarbon methods, which have an effective range of approximately 50,000 years. This meant that some of the material in the sediments was at least 50,000 years old, and possibly much older. Other organic material yielded radiocarbon ages within the datable range, suggesting deposition within the past few tens of thousands of years. The mixture of ages within a single sediment layer complicated the interpretation, as it suggested that the sediments contained material from multiple sources and multiple time periods, mixed together by subglacial processes. The team that analysed the biological remains in the sediments was led by David Harwood of the University of Nebraska-Lincoln, a micropaleontologist with decades of experience studying diatom assemblages in Antarctic sediments. Harwood and his colleagues identified diatom species in the Lake Mercer sediments that were characteristic of freshwater and brackish water environments, not the marine diatoms that dominate the open Southern Ocean, but species typically found in lakes, ponds, and estuaries. The freshwater character of the diatom assemblage was significant because it indicated that the organisms had lived in a landlocked or semi-enclosed body of water, not in the open ocean, consistent with the interpretation that they had inhabited a surface lake or pond at the site during an ice-free period. The crustacean remains were equally revealing. The exoskeletons were identified as belonging to small freshwater or brackish water species, consistent with the diatom evidence. Some of the fragments were well preserved, retaining details of their surface structure that allowed preliminary taxonomic identification. The preservation quality suggested that the organisms had not been transported long distances through the subglacial drainage system, which would have been expected to damage or destroy such delicate structures. Instead, the remains appeared to have been deposited relatively close to where the organisms had lived, consistent with local deposition in a surface aquatic environment. And it gets worse. The sediment cores also contained layers of organic-rich material, dark bands within the otherwise mineral-dominated stratigraphy that represented periods of enhanced biological productivity. These organic layers were interspersed with layers of glacial till, mineral-rich sediment deposited by ice flowing over bedrock. The alternation between organic layers and glacial layers suggested a cyclic pattern, a landscape that oscillated between ice-covered and ice-free conditions, with biological communities establishing themselves during ice-free periods and being buried by glacial sediment when the ice returned. This cyclic pattern was the most disturbing finding of all, because it implied that the retreat and advance of the ice sheet at this location was not a singular, catastrophic event, but a repeated phenomenon, a regular feature of the ice sheet's behavior over geological timescales. If the ice had retreated from this location once, it might be dismissed as an exceptional event, driven by unusual circumstances that might not recur. But if the ice had retreated and advanced multiple times, the implication was that the ice sheet's behavior in this region was inherently dynamic, prone to instability under natural climate forcing, and potentially responsive to the kind of warming that is now occurring as a result of anthropogenic greenhouse gas emissions. The core problem of the Lake Mercer discovery is therefore a problem of interpretation and implication. The biological remains in the sediments are real. Their presence has been confirmed by multiple independent analyses. The question is what they mean for the history and future of the West Antarctic ice sheet. The most conservative interpretation holds that the biological remains were deposited during the last interglacial, approximately 115,000 to 130,000 years ago, when global temperatures were 1 to 2 degrees warmer than pre-industrial levels and sea levels were 6 to 9 meters higher. During this period, the West Antarctic ice sheet may have partially or completely collapsed, exposing the bedrock beneath the ice to open water and terrestrial conditions. The organisms whose remains were found in the Lake Mercer sediments would have lived during this ice-free period, their bodies settling to the bottom of a shallow lake or pond that existed at the site before the ice returned. This interpretation is consistent with the broader body of evidence about last interglacial conditions in Antarctica. Multiple lines of evidence, from marine sediment cores, from ice cores, from Geological surveys of raised marine terraces around the Antarctic coast suggest that the West Antarctic ice sheet was significantly reduced during the last interglacial. The contribution of West Antarctic ice melt to last interglacial sea level rise has been estimated at 1.5 to 3.5 meters, which would require a substantial reduction in the ice sheet's extent. A more alarming interpretation holds that the ice-free conditions at the Lake Mercer site may have been more recent than the last interglacial. Some of the radiocarbon dates on organic material in the sediments fall within the range of the last 10 to 20,000 years, suggesting that the ice may have retreated from this location during or after the end of the last glacial period. If this interpretation is correct, the ice sheet in this region would have been absent much more recently than previously thought, and its subsequent re-advance to its current position would have occurred over a remarkably short geological timescale. This more recent timing would be deeply concerning for ice sheet stability. If the ice sheet retreated from the Lake Mercer site within the past 20,000 years and then re-advanced to its current position, it would imply that the ice sheet is capable of rapid retreat and re-advance over timescales of thousands to tens of thousands of years, much shorter than the hundreds of thousands of years typically associated with glacial interglacial cycles. It would suggest that the ice sheet is more sensitive to climate forcing than current models assume, and that the current warming trend could trigger a retreat that unfolds more quickly than predicted. But the interpretation of the radiocarbon dates is complicated by the possibility of contamination and reworking. Subglacial processes can transport organic carbon from one location to another, mixing material of different ages. Radiocarbon dating assumes that the dated material was deposited at the time the organisms lived and died. But if older organic carbon has been mixed into more recent sediments by subglacial water flow or ice sheet dynamics, the apparent ages of the material may not accurately reflect the timing of deposition. Sorting out the true chronology of the Lake Mercer sediments requires additional geochronological analyses, including exposure dating using cosmogenic nuclides, optically stimulated luminescence dating, and other methods that can independently constrain the age of the sediments and the timing of ice-free conditions. The challenge of dating the Lake Mercer sediments illustrates a broader problem in Antarctic geology. The ice sheet that covers the continent conceals the geological record from direct investigation, making it extraordinarily difficult to reconstruct the history of the landscape beneath the ice. Every piece of evidence must be extracted through expensive and technically demanding drilling operations. Every sample must be analysed with the awareness that subglacial processes may have mixed, transported, or altered the material, and every interpretation must contend with the possibility that the available evidence is incomplete, that the sediment cores recovered from a single borehole in a single lake may not be representative of the broader region or the full duration of the geological record. The core problem is that the Lake Mercer sediments tell a story that is simultaneously clear in its general outlines and deeply uncertain in its specific details. The general outline is clear. The ice was not always here. Organisms that require open water, sunlight, and terrestrial conditions once lived at this location. The ice sheet has retreated from this region in the geological past, and it may do so again if conditions are right. The specific details: when exactly did the ice retreat? How long was the ice-free period? How extensive was the retreat? How quickly did the ice return? Remain uncertain, subject to ongoing research and debate. Something is wrong with our assumptions about the permanence of the Antarctic ice. The ice sheet is not a monolith, unchanging and eternal. It is a dynamic system, responsive to climate forcing, capable of retreat and re-advance over geological timescales, and vulnerable to the same kinds of warming that are occurring today. The sediments at the bottom of Lake Mercer have provided the most direct evidence yet for this dynamism, evidence drawn not from distant marine cores or from theoretical models, but from the very bedrock over which the ice flows. But here's what makes this truly terrifying. The conditions that may have caused the ice to retreat in the past, warming temperatures and rising seas, are precisely the conditions that human activities are creating now. The warming that is currently underway, driven by the burning of fossil fuels and the release of greenhouse gases into the atmosphere, is pushing the climate system toward the same thresholds that may have destabilized the West Antarctic ice sheet during previous warm periods. The organisms in the Lake Mercer sediments lived in a world without ice at this location. If we continue on our current trajectory, the inhabitants of future centuries may live in that world again. The biological findings from Lake Mercer extended beyond the fossil remains in the sediments to include the living organisms in the lake water itself. Like Lake Williams before it, Lake Mercer contained an active, metabolizing microbial community sustained by chemical energy derived from rock-water interactions in the subglacial environment. The cell concentrations in the Lake Mercer water were comparable to those found at Lake Williams, on the order of tens of thousands of cells per milliliter, confirming that the subglacial biosphere extends across a significant portion of the Williams-Mercer ice stream catchment. The composition of the microbial community in Lake Mercer showed both similarities and differences compared to Lake Williams. The dominant metabolic groups, iron oxidizers, sulfur cyclers, and chemolethal autotrophs, were present in both lakes, as expected given the similar geological and chemical conditions at the two sites. But the relative abundances of different organisms varied between the two lakes, reflecting local differences in water chemistry, sediment composition, and flow patterns. Some organisms found in Lake Mercer had not been detected in the Lake Williams samples, suggesting that the microbial diversity of the subglacial environment exceeds what a single sampling site can reveal. The coexistence of living microbes and ancient biological remains in the same environment created a unique scientific opportunity. The living organisms represented the current state of the subglacial ecosystem, adapted to the dark, cold, pressurized conditions beneath the ice. The fossil remains represented past states of the environment, snapshots of ecosystems that had existed at the same location under radically different conditions. By comparing the two, researchers could trace the biological history of the site from an ice-free environment supporting photosynthetic algae and grazing crustaceans to the current subglacial environment supporting only chemolethal-autotrophic bacteria and archaea. The transition between these two states, from an open water, light-receiving ecosystem to a sealed, dark, chemically-driven ecosystem, represents one of the most dramatic environmental transformations imaginable. The organisms that lived at the site during its ice-free period depended on sunlight for their primary energy source. When the ice returned and sealed the environment from the surface, sunlight was eliminated and the entire energy basis of the ecosystem was destroyed. Every photosynthetic organism died. Every animal that depended on photosynthetic productivity for food died. The only organisms that survived were those capable of deriving energy from chemical reactions rather than light. The chemolethal autotrophs that now dominate the subglacial community. This biological catastrophe, the annihilation of an entire ecosystem and its replacement by a fundamentally different one, occurred at this site at least once and possibly multiple times as the ice sheet advanced and retreated in response to climate forcing. Each advance of the ice extinguished the photosynthetic community and established a chemolithal-autotrophic one. Each retreat of the ice extinguished the chemolithal-autotrophic community, or at least marginalized it, and established a photosynthetic one. The sediments preserved a record of these oscillations, with organic-rich layers representing ice-free periods and mineral-rich layers representing glacial periods alternating through the core. The ancient DNA extracted from the sediments added further detail to this picture. Using advanced DNA extraction and sequencing techniques, the SALSA team recovered genetic material from organisms that had lived at the site during ice-free periods. The DNA was degraded, as expected for material that was potentially thousands of years old, but enough sequence information survived to allow preliminary identification of some of the organisms. The DNA results confirmed the microscopic identifications of crustaceans and diatoms, and revealed additional organisms that were not preserved as recognizable physical remains, including species of mosses, fungi, and various groups of bacteria associated with surface freshwater environments. The presence of moss DNA was particularly significant. Mosses are terrestrial plants that grow on land surfaces exposed to the atmosphere. Their presence in the sediments implied not merely ice-free conditions, but the existence of exposed land surfaces, soil and vegetation at the site. The landscape during ice-free periods was therefore not merely a shallow lake surrounded by bare rock. It was a vegetated landscape, with moss-covered ground bordering a shallow body of water in a climate warm enough to support plant growth, in a region that is now buried beneath more than a kilometre of ice. The picture that emerged from the combined evidence from the diatoms, the crustaceans, the tardigrades, the mosses, the ancient DNA, the sediment chemistry, was of an Antarctic landscape that had undergone multiple cycles of transformation, from a vegetated, aquatic, sunlit environment to a sealed, dark, frozen environment, and back again. Each cycle was driven by the advance and retreat of the ice sheet, and each cycle left its mark in the sediments, a geological diary written by the organisms that lived and died at the site over the course of the ice sheet's oscillations. The scientific significance of this diary cannot be overstated. The Lake Mercer sediments represent the first direct record of ice-free conditions recovered from beneath the current extent of the West Antarctic ice sheet. Previous evidence for past ice sheet retreat had been indirect, inferred from marine sediment cores, from raised beach deposits, from ice core chemistry, from modelling studies. The Lake Mercer sediments provided something that none of these indirect sources could offer: a record from the very location where the ice currently sits, showing what the environment looked like when the ice was absent. This directness is scientifically powerful because it eliminates many of the uncertainties associated with indirect evidence. Marine sediment cores record conditions at the ocean floor, far from the ice margin, and interpreting their implications for the ice sheet itself requires models and assumptions that may introduce errors. Ice core records are limited in their geographic coverage and temporal extent. Modelling studies depend on the accuracy of the models, which are constrained by our incomplete understanding of ice sheet dynamics. The Lake Mercer sediments bypass all of these limitations by providing a direct in situ record of the conditions that prevailed at the ice-bedrock interface during different phases of the ice sheet's history. The implications for ice sheet modelling are significant. Climate models that simulate the behaviour of the West Antarctic ice sheet must reproduce its observed past behaviour in order to be trusted to predict its future behaviour. If the models cannot reproduce the ice-free conditions implied by the Lake Mercer sediments, under the climate forcing conditions that prevailed during the time periods when those sediments were deposited, the models are missing something, some process, some feedback, some mechanism that allows the ice sheet to retreat further and faster than the models predict. This discrepancy between the observational evidence and the model predictions is the core of the problem. Many current ice sheet models predict that the West Antarctic ice sheet is relatively stable under modest warming, retreating slowly and incrementally in response to rising temperatures and warming ocean waters. The Lake Mercer sediments suggest that the reality may be different, that the ice sheet has retreated rapidly and completely from this region under natural climate forcing that was, in many cases, less intense than the forcing that anthropogenic warming is now producing. If the models are underestimating the ice sheet's sensitivity to climate forcing, the projections of future sea level rise derived from those models may also be underestimates. The political and societal implications of this possibility are enormous. Sea level rise projections are used to plan infrastructure investments, set building codes, design flood defences, and make decisions about the habitability of coastal areas around the world. If the West Antarctic ice sheet is more sensitive to warming than current models assume, the timelines for sea level rise may be shorter, the magnitudes may be larger, and the consequences for coastal communities may be more severe than current planning scenarios anticipate. The Lake Mercer sediments, retrieved from beneath a kilometre of ice by a team of scientists working in one of the most hostile environments on Earth, have implications that extend to the lives and livelihoods of billions of people who live in coastal regions around the world. The core problem of the Lake Mercer discovery is therefore not merely a scientific problem. It is a societal problem, a challenge that connects the paleontological record of a subglacial lake to the economic, political, and humanitarian dimensions of climate change. The organisms in the sediments, the crustaceans and diatoms and tardigrades and mosses that lived at this site during ice-free periods are messengers from a past that may also be our future. Their presence tells us that the ice can go away, that the landscape beneath the ice can be exposed, that the conditions for open water and terrestrial ecosystems can exist in a region that is currently covered by the densest, deepest ice on the West Antarctic ice sheet. The question is not whether the ice can retreat. The sediments prove that it has. The question is when it will retreat again, how quickly, and what the consequences will be for a civilization that has built its coastal infrastructure on the assumption that the great ice sheets of the polar regions are permanent features of the planetary landscape. The debate over the age and origin of the biological remains in the Lake Mercer sediments has been one of the most active areas of research in Antarctic science since the SALSA's results were first presented to the scientific community. Several competing hypotheses have been proposed, each with different implications for ice sheet history. The reworking hypothesis suggests that the biological remains were not deposited at the Lake Mercer site during an ice-free period, but were instead eroded from older deposits elsewhere and transported to the lake by subglacial water flow. Under this hypothesis, the crustacean shells, diatom frustules, and other biological remains could have originated in marine or freshwater sediments deposited millions of years ago, long before the current glacial period, and been carried into the lake by the active subglacial drainage system. This hypothesis is appealing in its simplicity, but it faces several objections. The preservation quality of the biological remains, particularly the crustacean exoskeletons, is higher than would be expected for material that has been transported long distances through turbulent subglacial channels. Delicate biological structures tend to be damaged or destroyed by transport, and the relatively intact state of the Lake Mercer remains suggests that they were deposited relatively close to where the organisms lived. The in situ deposition hypothesis suggests that the organisms lived and died at or near the current location of Lake Mercer during a period when the ice sheet was absent from this region. This is the interpretation that carries the most dramatic implications for ice sheet history, because it requires the West Antarctic ice sheet to have retreated from this location entirely, exposing the bedrock to open water and terrestrial conditions. The dating of this ice-free period is the central challenge, with evidence pointing to the last interglacial as the most likely candidate, but with some data suggesting more recent ice-free conditions. A third hypothesis, the ice shelf cavity hypothesis, offers a middle ground. Under this interpretation, the organisms lived in a marine environment, in the water cavity beneath a floating ice shelf that covered the region during a period of reduced ice sheet extent. Ice shelves, the floating extensions of the ice sheet that cover large areas of the Ross Sea and other Antarctic coastal regions, create environments where marine organisms can live in relative darkness beneath hundreds of meters of floating ice. If the West Antarctic ice sheet retreated sufficiently to allow a floating ice shelf to cover the Lake Mercer region, marine organisms including diatoms and small crustaceans could have lived in the subshelf environment, and their remains could have settled to the seafloor where they were subsequently buried by sediment when the ice sheet re-advanced and grounded over the site. This hypothesis is consistent with the geomorphological evidence from the broader region. Studies of the seafloor beneath the Ross Ice Shelf have revealed features consistent with marine conditions, including biological communities that currently live in the dark, cold water beneath the floating ice. The subshelf environment supports a reduced but functional marine ecosystem, sustained by nutrients delivered by ocean currents flowing beneath the ice shelf. If a similar environment existed at the Lake Mercer site during a period of ice sheet retreat, it could account for the biological remains in the sediments, without requiring the complete removal of ice from the region. Each of these hypotheses has testable predictions, and the ongoing analysis of the Lake Mercer sediments is designed to discriminate among them. If the biological remains were reworked from older deposits, their ages should be mixed and inconsistent, with material from multiple time periods jumbled together in the same sediment layer. If they were deposited in situ during an ice-free period, their ages should cluster around a specific time period, and the sediment layers should show a clear transition from glacial to ice-free to glacial conditions. If they were deposited in a sub-ice shelf marine environment, the diatom and crustacean species should be marine rather than freshwater, and the sediment chemistry should show signatures of seawater rather than freshwater. The initial results are ambiguous, with some evidence supporting each hypothesis and no single hypothesis able to account for all of the observed data. The diatom assemblage includes both freshwater and marine species, suggesting that the environment may have transitioned between freshwater and marine conditions at different times, or that the sediments contain material from multiple different periods and environments. The radiocarbon ages of the organic material span a wide range, consistent with either mixing of material from different time periods or deposition over an extended period, and the sediment chemistry shows signatures that are intermediate between glacial and non-glacial conditions, resisting simple classification. The resolution of this debate will require additional data from additional sites. A single set of sediment cores from a single subglacial lake cannot, by itself, resolve questions about the behavior of an ice sheet that covers nearly 2 million square kilometers. Sediment cores from other subglacial lakes in the region, from different positions along the ice sheet's flow path and at different distances from the coast, would provide the spatial coverage needed to reconstruct the ice sheet's retreat pattern and determine whether the retreat was uniform across the region or confined to specific locations. The SOLSAR team has recognized this need and has proposed follow-up missions to additional subglacial targets in the West Antarctic region. But each mission requires years of planning, millions of dollars in funding, and the mobilization of a large multidisciplinary team to one of the most logistically challenging environments on Earth. The pace of Antarctic sediment research is measured in decades, not years, and the complete picture of ice sheet history that the Lake Mercer sediments hint at may not emerge for a generation or more. In the meantime, the organisms in the sediments remain where they were found, preserved in their layers of mud beneath a kilometre of ice, a biological archive of past conditions waiting to be more fully read. The crustacean shells and diatom frustules are patient witnesses, their testimony frozen in sediment, their message clear in its general outlines if uncertain in its specific details. The ice was not always here. Life once flourished at this location, in conditions very different from those that prevail today, and the forces that removed the ice in the past are cousins of the forces that threaten the ice today. The core problem of Lake Mercer sits at the intersection of biology, geology, and climate science, a nexus where the remains of tiny organisms become evidence in a high-stakes debate about the future of the planet's ice sheets and the stability of its coastlines. It is a problem that cannot be solved by any single discipline or any single mission. It demands the combined efforts of paleontologists and glaciologists, geochemists and climate modelers, field scientists and laboratory analysts, working together over years and decades to extract the full meaning from a few centimeters of mud recovered from one of the most inaccessible places on Earth. The stakes could not be higher. The biological remains in the Lake Mercer sediments are not merely scientific curiosities. They are warning signs, evidence that the ice sheet we depend upon for coastal stability has not always been stable, that it has responded dramatically to climate forcing in the past, and that it may respond dramatically again in the future. Reading these warning signs correctly, understanding what they tell us about the thresholds and timescales of ice sheet retreat, is one of the most urgent tasks facing the scientific community today. There is an additional dimension to the core problem that connects the biological discoveries at Lake Mercer to the brorsaurozory of carbon cycling beneath the Antarctic ice sheet. The organic carbon found in the sediments The ancient remains of organisms that once lived in open water conditions at the site represents a stored reservoir of biological energy, locked in sedimentary deposits beneath the ice for thousands or possibly hundreds of thousands of years. This ancient organic carbon is not inert. It is being consumed by the living microbes in the current subglacial environment, broken down and metabolized by organisms that derive energy and nutrients from it. The consumption of ancient organic carbon by subglacial microbes has implications that extend beyond the local ecosystem. When microbes consume organic carbon, they release carbon dioxide and methane as metabolic byproducts. These gases dissolve in the subglacial water, or, in the case of methane, may accumulate as methane hydrates in the sediments. If the subglacial water is eventually discharged into the Southern Ocean through the subglacial drainage system, or through the grounding line where the ice sheet lifts off the bedrock, the dissolved carbon dioxide and methane it carries represent a transfer of ancient carbon from the subglacial reservoir to the ocean and potentially to the atmosphere. The magnitude of this carbon flux is unknown but could be significant. The sediments beneath the West Antarctic ice sheet contain substantial quantities of organic carbon deposited by marine and terrestrial ecosystems during pre-glacial and interglacial periods. If subglacial microbes are actively consuming this carbon and releasing greenhouse gases as a result, the subglacial biosphere could be a source of atmospheric carbon dioxide and methane that is not currently accounted for in global carbon budgets. The contribution would be small compared to anthropogenic emissions, but it could be significant over geological timescales, and it could increase as ice sheet retreat exposes more organic-rich sediments to microbial activity. The methane dimension is particularly concerning. Methane is a greenhouse gas roughly 80 times more potent than carbon dioxide over a 20-year time horizon. If the sediments beneath the West Antarctic ice sheet contain substantial quantities of methane, either as dissolved gas in subglacial water or as solid methane hydrates in the sediments, the release of that methane during ice sheet retreat could create a positive feedback loop, in which ice retreat releases methane, which causes additional warming, which causes further ice retreat, which releases more methane. This feedback loop is analogous to the permafrost carbon feedback that has been identified in the Arctic, where thawing permafrost releases stored carbon as greenhouse gases, amplifying the warming that caused the thawing in the first place. The Lake Mercer sediments provide evidence that both carbon dioxide and methane are produced by microbial metabolism in the subglacial environment. The geochemical analyses of the lake water revealed dissolved methane at concentrations consistent with active microbial methanogenesis, the biological production of methane from carbon dioxide and hydrogen. The presence of methanogenic microbes in the community confirms that the biological machinery for methane production is present and functioning in the subglacial environment. The interaction between ancient carbon, living microbes, greenhouse gas production, and ice sheet dynamics creates a system of feedbacks and connections that the Lake Mercer discovery has brought into sharp focus. The organisms that once lived at this site during ice-free periods deposited organic carbon in the sediments. That organic carbon is now being consumed by subglacial microbes producing greenhouse gases. Those greenhouse gases may be released when the ice sheet retreats, contributing to further warming and further ice retreat. And the retreat of the ice sheet exposes more organic-rich sediments to microbial activity, accelerating the cycle. This nested system of feedbacks is poorly understood and difficult to model. The rates of microbial carbon consumption in the subglacial environment are not well constrained. The volumes of methane and carbon dioxide stored beneath the ice sheet are uncertain. The pathways by which these gases might reach the atmosphere through subglacial water discharge, through cracks in the ice, through the melting of methane hydrates, are not fully mapped, and the interaction between these biological and chemical processes and the physical dynamics of ice sheet retreat is a frontier area of research that sits at the boundary of multiple scientific disciplines. The Lake Mercer discovery has illuminated these connections in a way that no previous Antarctic research project had achieved. by recovering both living microbes and ancient biological remains from the same environment. The SALSA team provided evidence that the subglacial carbon cycle is active, dynamic, and connected to both the geological past and the climate future. The crustacean shells and diatom frustules in the sediments are not merely evidence of past ice-free conditions. They are evidence of past carbon deposition, the raw material from which the current subglacial ecosystem derives part of its energy, and from which future greenhouse gas emissions may be generated. The core problem of Lake Mercer is ultimately a problem about time, about the connection between past, present, and future. The past is written in the sediments, in the biological remains and chemical signatures of environments that no longer exist. The present is sustained by the metabolism of living microbes, consuming the carbon deposited by past ecosystems and producing the greenhouse gases that may shape future climates. And the future is contingent, dependent on the choices that human civilization makes about greenhouse gas emissions, dependent on the response of the ice sheet to the warming those emissions cause, and dependent on the feedbacks between biological, chemical, and physical processes that the Lake Mercer discovery has only begun to reveal. The Lake Mercer discovery was not made in isolation. It emerged from a decades-long program of Antarctic exploration that has brought multiple teams, multiple nations, and multiple technologies to bear on the challenge of understanding what lies beneath the ice. The SALSA results fit into a broader scientific landscape shaped by previous successes, previous failures, and ongoing efforts to extend the reach of subglacial science to new sites and deeper targets. The first angle of approach is the one that directly preceded SALSA, the Wizard project at Lake Whillans. The clean access to Lake Whillans in 2013 was the proof of concept that made SALSA possible. Without Wizard, the scientific community would not have known that clean subglacial access was achievable, and the funding agencies would not have had the confidence to invest in a follow-up mission. The Wizard results demonstrated that living microbial communities existed in the subglacial environment, that hot water drilling could provide clean access to shallow subglacial lakes, and that the contamination control protocols developed by the team were adequate for biological sampling. But Wizard also revealed the limitations of sampling a single site. The Lake Whillans community represented one point in a vast and heterogeneous system, and the scientific community recognized that additional samples from additional sites were needed to determine how representative the Whillans findings were. SALSA was designed to address this gap by accessing a second subglacial lake in the same region, at a slightly greater depth and in a slightly different geological and hydrological setting. The comparison between the two sites would provide the first opportunity to assess spatial variability in the subglacial biosphere, testing whether the organisms and metabolic processes found at Whillans were also present at Mercer. The results confirmed that the basic features of the subglacial ecosystem, the chemolitho-autotrophic metabolism, the iron and sulfur cycling, the presence of active microbial communities, were indeed present at both sites. But the sediment findings at Lake Mercer added an entirely new dimension to the investigation, one that Wizard had not encountered. The shallow sediments at Lake Whillans had not contained the rich biological remains found at Lake Mercer, possibly because the geological setting of the two sites differed, or because the sediment cores recovered at Whillans did not penetrate deep enough to reach layers deposited during ice-free periods. The Lake Mercer sediments, by contrast, preserved a record that extended further back in time and included evidence of environmental conditions that had no parallel in the Whillans data. The second angle of approach is the broader effort to understand the stability of the West Antarctic ice sheet through field observations, modelling, and remote sensing. The Thwaites Glacier Research Program, a joint United States-United Kingdom initiative known as the International Thwaites Glacier Collaboration, has been conducting extensive studies of the Thwaites Glacier system since 2018. The Thwaites program has deployed autonomous vehicles beneath the glacier, drilled through the ice to access the ocean cavity beneath it, and conducted detailed mapping of the sea floor over which the glacier is retreating. The results have confirmed that warm ocean water is reaching the base of the glacier, melting it from below and accelerating its retreat. The Thwaites research is directly relevant to the Lake Mercer story because it provides the modern context for the paleoclimate evidence in the Mercer sediments. If the West Antarctic ice sheet retreated from the Lake Mercer region in the geological past, the retreat likely began at the margins of the ice sheet, where warm ocean water first penetrated beneath the ice shelves and propagated inland along the ice streams, including the Mercer ice stream, as the grounding line retreated. The processes that are currently observed at Thwaites, warm water penetrating the ice-ocean interface, grounding line retreat on a downward sloping bed, accelerating ice flow, are the same processes that likely drove the past retreat recorded in the Lake Mercer sediments. The modern observations at Thwaites and the paleoclimate evidence at Lake Mercer are two views of the same phenomenon, one observed in real time and the other reconstructed from geological archives. The third angle of approach involves the marine geological record from the Ross Sea and surrounding ocean basins. The Andril program, which drilled sediment cores from beneath the McMurdo Ice Shelf in 2006 and 2007, recovered a record spanning the past 13 million years that showed repeated cycles of ice sheet advance and retreat. The Andril cores contained layers of glacial sediment deposited during periods when the ice sheet was grounded over the drill site, alternating with layers of marine sediment deposited during periods when the ice had retreated and the site was covered by open ocean. The pattern of alternation demonstrated that the West Antarctic ice sheet had collapsed and reformed dozens of times over the past several million years, each cycle driven by changes in global climate and ocean circulation. The Andril results provided the long-term context that the Lake Mercer sediments filled in at shorter timescales, where Andril showed that the ice sheet had oscillated over millions of years. The Lake Mercer sediments showed that similar oscillations had occurred within the most recent glacial-interglacial cycles, potentially within the past 120,000 years or less. The convergence of long-term and short-term evidence created a consistent picture of an ice sheet that is fundamentally dynamic, prone to retreat under warm conditions and re-advance under cold conditions, with the biological remains in the sediments serving as markers of the ice-free periods. The fourth angle of approach is the satellite-based monitoring of the Antarctic ice sheet, which has transformed our ability to observe ice sheet changes in real time. The GRACE and GRACE-FO satellite missions, which measure the Earth's gravitational field with sufficient precision to detect changes in ice mass, have documented a steady acceleration in the rate of ice loss from the West Antarctic ice sheet. Between 2002 and 2020, the West Antarctic ice sheet lost approximately 3,100 gigatons of ice, contributing approximately 9 millimeters to global sea level rise. The rate of ice loss has been accelerating, with recent years showing significantly higher losses than the earlier part of the observation period. The numbers are striking. The West Antarctic ice sheet is currently losing ice at a rate of roughly 150 gigatons per year, equivalent to approximately 0.4 millimeters of sea level rise per year from this source alone. While this may seem small, the trend is accelerating, and the potential for rapid, non-linear increases in ice loss as the Thwaites Glacier and other major outlets retreat makes the current rate a poor guide to future behavior. The Lake Mercer sediments suggest that past retreats of the ice sheet may have been much faster than the current rate of change, with the ice sheet potentially collapsing from its current configuration to a significantly reduced state over timescales of centuries to millennia rather than tens of thousands of years. The satellite observations also reveal the dynamic behavior of the subglacial hydrological system that connects Lake Mercer to the broader ice stream catchment. Interferometric synthetic aperture radar measurements from satellite platforms have detected the elevation changes on the ice surface that indicate subglacial lake filling and draining events. These observations have been used to track the water budget of individual subglacial lakes, including Lake Mercer, and to map the drainage pathways that connect the lakes to each other and to the coast. The hydrological data provides the context needed to understand how the subglacial biosphere is connected across the ice sheet and how changes in ice dynamics might alter the flow of water, nutrients, and organisms through the subglacial drainage network. The fifth angle of approach involves the development of new analytical techniques that are being applied to the Lake Mercer samples in an ongoing program of laboratory research. Since the initial results were published, the SALSA team and its collaborators have been subjecting the sediment cores and water samples to increasingly sophisticated analyses, seeking to extract additional information about the timing, duration, and environmental conditions of the ice-free periods recorded in the sediments. Cosmogenic nuclide dating, which measures the accumulation of rare isotopes produced by cosmic ray bombardment of rock surfaces, is being applied to mineral grains in the sediments to determine how long the bedrock at the Lake Mercer site has been exposed to the atmosphere. When the ice sheet covers a site, cosmic rays cannot reach the bedrock and the production of cosmogenic nuclides ceases. When the ice retreats and the bedrock is exposed, cosmic ray bombardment resumes and nuclides begin to accumulate. By measuring the concentration of cosmogenic nuclides in the sediment grains, researchers can estimate the total duration of exposure, providing an independent constraint on the timing and extent of ice-free conditions. Optically stimulated luminescence dating, which measures the time since sediment grains were last exposed to sunlight, is being applied as a complementary geochronological technique. When sediment grains are buried and shielded from light, they accumulate a radiation dose from natural radioactivity in the surrounding material. When the grains are exposed to light, the accumulated dose is reset. By measuring the accumulated dose in sediment grains from the Lake Mercer cores, researchers can determine when the grains were last exposed to sunlight, providing another line of evidence about the timing of ice-free conditions. These analytical techniques take months to years to produce results, and the full geochronological framework for the Lake Mercer sediments is still being developed. The initial publications from the SALSA team presented preliminary age estimates based on radiocarbon dating, but the more advanced techniques are expected to provide tighter constraints on the timing of ice-free conditions and to resolve some of the ambiguities in the radiocarbon data. The ongoing analysis of the Lake Mercer samples also includes detailed studies of the microbial community in the lake water and sediments, using methods that have advanced significantly since the initial Wissard analyses. Metatranscriptomic analysis, which measures the genes that are actively being expressed by the microbial community rather than merely identifying the organisms present, is providing new insights into the metabolic activities of the subglacial ecosystem. These analyses reveal which organisms are most metabolically active, which genes are being expressed under the specific conditions of the subglacial environment, and how the community's metabolic priorities differ from those of surface microbial communities. Stable isotope analysis of the dissolved compounds in the lake water and sediment pore water is providing information about the sources and cycling of carbon, nitrogen, sulfur, and other elements in the subglacial environment. The isotopic ratios of these elements serve as tracers of the metabolic processes that have transformed them, allowing researchers to quantify the rates of microbial carbon fixation, methane production, sulfur cycling, and other key biogeochemical processes. Together, these multiple angles of approach, from field observations to satellite monitoring, from marine sediment records to subglacial sediment analysis, from microbiology to geochronology, are converging on a comprehensive understanding of the Lake Mercer system and its implications for ice sheet history and climate science. The picture that is emerging is more complex, more dynamic, and more consequential than anyone anticipated when the SALSA project was first conceived. Each new result adds detail to the picture, and each new detail raises additional questions about the behavior of the ice sheet, the resilience of the subglacial biosphere, and the vulnerability of both to the warming that is underway. The pattern of exploration around Lake Mercer reflects a broader truth about Antarctic science, that understanding the frozen continent requires patience, investment, and the willingness to pursue multiple lines of evidence simultaneously. No single mission, no single technique, and no single discipline can answer the questions that Antarctica poses. The answers emerge gradually from the convergence of many different types of evidence gathered over many years by many different teams. The Lake Mercer discovery is one piece of a much larger puzzle, and fitting it into the broader picture of ice sheet dynamics and global climate change is a task that will occupy the scientific community for years, if not decades, to come. The connection between the Lake Mercer findings and the search for life on other worlds provides another critical angle of exploration that deserves attention in this section. The SALSA results extended the Wizard proof of concept in an important direction. They demonstrated not only that life exists in the subglacial environment, but that the subglacial environment preserves a biological record of past conditions that can be read using standard paleontological and geochemical techniques. This dual capability, the ability to detect both living organisms and their ancient predecessors, is directly relevant to the design of future missions to the icy moons of the outer solar system. The Europa Clipper mission, launched in 2024 and scheduled to arrive at Jupiter in 2030, will assess the habitability of Europa's subsurface ocean by studying the moon's ice shell, surface chemistry, and geology from orbit. The mission is not designed to land on Europa or penetrate its ice shell, but the data it collects will inform the design of subsequent missions that may attempt to access the ocean beneath the ice, using technologies descended from the drilling and probing systems developed for Antarctic subglacial exploration. The Lake Mercer results are relevant to Europa mission design because they demonstrate what kinds of evidence can be preserved in the sediments of an ice-sealed aquatic environment. If Europa's ocean floor contains sediments, and if those sediments preserve biological remains analogous to the fossils found in the Lake Mercer cores, then a future mission equipped with the right instruments could potentially detect evidence of past or present biological activity on Europa without needing to find living organisms in real time. The detection of fossil biosignatures, preserved biological structures, or chemical signatures in ancient sediments would be as significant as the detection of living cells, because it would demonstrate that biological processes had occurred in Europa's ocean at some point in its history. This approach to biosignature detection, looking for the traces that life leaves in its environment, rather than looking for living organisms directly, has been a central strategy in the Mars exploration program. The Perseverance rover, which landed on Mars in 2021, is specifically designed to collect rock and sediment samples that might contain preserved biosignatures from Mars's ancient, wetter past. The strategy is based on the recognition that finding living organisms on another world is extraordinarily difficult, while finding the traces that past organisms left in the geological record may be more achievable. The Lake Mercer sediments demonstrate that this approach works on Earth, in an environment analogous to those that may exist on other worlds. The technology development stimulated by the SALSA mission has also advanced the state of the art in several areas relevant to planetary exploration. The miniaturized sensors used to monitor contamination in the drilling water, the sample collection instruments designed for deployment through narrow boreholes, the analytical protocols for distinguishing indigenous organisms from contaminants, all of these have potential applications in the design of instruments for future planetary missions. The experience of operating complex scientific equipment in the extreme conditions of the Antarctic ice sheet has provided valuable lessons for the engineers who will design instruments for the even more extreme conditions of an ice-penetrating mission to Europa or Enceladus. The Enceladus connection is equally significant. Saturn's small moon Enceladus has been identified as one of the most promising targets in the solar system for the detection of extraterrestrial life, because the plumes of water vapor erupting from its south pole provide a natural sampling mechanism that eliminates the need to drill through the ice. A future mission could fly through the plumes and analyze their composition, looking for the chemical signatures of biological activity. The SALSA results are relevant because they provide a benchmark for the kinds of biological signatures that might be detectable in such a plume analysis. The metabolic products of chemolitho-autotrophic organisms, the organic molecules produced by microbial carbon fixation, and the isotopic signatures of biological element cycling. The intersection of Antarctic subglacial science and planetary exploration is one of the most productive interdisciplinary collaborations in modern science. Each field informs the other, with Antarctic discoveries shaping the questions and methods of planetary exploration, and planetary exploration motivating new investigations of Antarctic subglacial environments. The Lake Mercer discovery has strengthened this collaboration by demonstrating that ice-sealed environments on Earth can preserve both living organisms and their fossil predecessors, providing a richer and more complex biological record than had previously been appreciated. The practical challenge of extending the Lake Mercer results to other sites in Antarctica remains formidable. Each subglacial access mission requires years of planning, millions of dollars in funding, and the mobilization of a large team to a remote location in one of the most hostile environments on Earth. The limited number of missions that can be conducted in any given Antarctic season, constrained by logistics, budget, and the availability of trained personnel, means that the exploration of the subglacial biosphere proceeds at a pace far slower than the urgency of the scientific questions would justify. Several approaches have been proposed to accelerate the pace of discovery. One approach is to develop lighter, more portable drilling systems that can be deployed more quickly and at lower cost than the large hot water systems used by Wizard and Salsa. Smaller drilling systems, capable of penetrating hundreds of meters of ice rather than thousands, could be used to access shallow subglacial water bodies and sediments at multiple sites across Antarctica in a single season, providing the spatial coverage needed to assess the variability of the subglacial environment. Another approach is to exploit existing boreholes and natural access points. Ice shelf boreholes, drilled through floating ice to access the ocean cavity beneath, provide access to marine-influenced subglacial environments at the margins of the ice sheet. Glacial moulins, vertical channels created by surface meltwater draining through the ice, provide natural conduits to the base of the ice sheet in some locations, though these features are rare in Antarctica compared to Greenland, and the boreholes drilled for ice core research at deep interior sites like Dome C, Dome A, and the South Pole could potentially be extended to reach the subglacial environment beneath these sites, though the extreme depths involved present significant technical challenges. The development of autonomous monitoring systems represents a third approach. Instead of conducting expensive, time-limited drilling campaigns that provide snapshots of the subglacial environment at a single moment in time, autonomous instruments deployed at the ice-bedrock interface could monitor conditions continuously over months or years, capturing the temporal variability that single-visit campaigns inevitably miss. Seismometers, temperature sensors, and chemical analyzers deployed through boreholes and left in place after the drilling team departs could provide continuous data streams transmitted to the surface through fiber optic cables or acoustic links that would transform our understanding of subglacial dynamics. Each of these approaches has merits and limitations, and the optimal strategy for advancing subglacial science will likely involve a combination of all of them. The heavy investment missions like SALSA, capable of recovering pristine samples from deep subglacial targets, will continue to provide the highest quality data and the most dramatic discoveries, but they must be complemented by lighter, faster, more widely distributed observations that fill in the gaps between the major missions and provide the spatial and temporal context needed to interpret the high-resolution data from individual sites. The story of the explorations and attempts that surround the Lake Mercer discovery is, at its core, a story about the relationship between ambition and capability. The ambition of the scientific community is vast: to understand the subglacial biosphere, to reconstruct the history of the Antarctic ice sheet, to predict its future behavior, and to apply the lessons learned to the search for life on other worlds. The capability is growing but still limited, constrained by budgets, by logistics, by technology, and by the fundamental difficulty of working in one of the most hostile environments on the planet. The gap between ambition and capability is the space within which the next generation of discoveries will be made. Every advance in drilling technology, every improvement in analytical methods, every new satellite instrument, every additional funding commitment narrows the gap. And the discoveries at Lake Whillans and Lake Mercer, the proof that life exists beneath the ice and that the ice has retreated in the geological past, have provided the motivation and the justification for closing it. The role of ice sheet modelling in interpreting the Lake Mercer findings deserves particular attention because models are the primary tools through which the scientific community translates paleoclimate evidence into predictions about future ice sheet behaviour. The field of ice sheet modelling has advanced dramatically in the past 2 decades, driven by the availability of satellite observations, increased computational power, and improved understanding of the physical processes that govern ice flow. Modern ice sheet models simulate the behaviour of the Antarctic ice sheet by solving equations that describe the flow of ice under the influence of gravity, the interaction between the ice and the ocean at the grounding line, the effects of surface temperature and snowfall on ice mass balance, and the role of subglacial water in lubricating the ice-bedrock interface. These models can be run forward in time, predicting the ice sheet's response to specified climate scenarios, or backward in time, reconstructing past ice sheet configurations from known climate histories. The Lake Mercer sediments provide a critical test for these models. If the sediments record ice-free conditions at the Lake Mercer site during the last interglacial, models should be able to reproduce this retreat when driven with last interglacial climate forcing. If the models cannot reproduce the observed retreat, they are missing a process or mechanism that is important for the ice sheet's sensitivity to climate change. Identifying and incorporating the missing mechanism would improve the model's ability to predict future ice sheet behaviour and provide more reliable projections of sea level rise. Several studies have attempted to simulate the behaviour of the West Antarctic ice sheet during the last interglacial, with mixed results. Some models predict significant retreat of the ice sheet under last interglacial conditions, consistent with the Lake Mercer evidence. Others predict more modest changes, with the ice sheet remaining largely intact even under warmer temperatures. The discrepancies between models reflect differences in how they represent key processes, particularly the interactions between the ice and the ocean, the response of ice flow to changes in subglacial water, and the dynamics of ice shelf collapse. The mechanism of marine ice cliff instability, proposed by researchers in the mid-2010s, has been a particularly contentious topic in the modelling community. This mechanism posits that if an ice shelf collapses entirely, exposing a tall ice cliff at the edge of the ice sheet, the cliff may be structurally unstable and collapse under its own weight. The collapse would expose a new ice cliff further inland, which would also collapse, creating a chain reaction of cliff failure that could cause the rapid disintegration of the ice sheet. If marine ice cliff instability operates as proposed, it could accelerate ice sheet retreat dramatically, potentially contributing several meters to sea level rise within a few centuries. The evidence for marine ice cliff instability is debated. Some models that include this mechanism predict rapid ice sheet collapse under modest warming scenarios, consistent with the scale of ice retreat implied by the Lake Mercer sediments. Other models that omit the mechanism predict more gradual retreat. The Lake Mercer sediments cannot directly confirm or refute the existence of marine ice cliff instability, but they provide a boundary condition that models must satisfy. Whatever mechanisms they include, they must be able to produce ice-free conditions at the Lake Mercer site under the climate forcing that prevailed during past warm periods. The modelling efforts are further complicated by the role of the Ronne Ice Shelf, the enormous slab of floating ice that currently buttresses the ice streams draining the West Antarctic ice sheet toward the Ross Sea. The Ronne Ice Shelf acts as a brake on ice flow, slowing the discharge of ice from the interior of the ice sheet to the ocean. If the Ronne Ice Shelf were to thin significantly or collapse, the ice streams it buttresses, including the Mercer ice stream, would accelerate dramatically, increasing the rate of ice loss and potentially triggering the kind of retreat recorded in the Lake Mercer sediments. The stability of the Ronne Ice Shelf is therefore central to the question of whether the Lake Mercer region will experience ice-free conditions again in the future. Current observations show that the Ronne Ice Shelf is relatively stable compared to the more vulnerable ice shelves of the Antarctic Peninsula and the Amundsen Sea sector, but it is not immune to the warming of the ocean waters that circulate beneath it. Models project that continued ocean warming could eventually undermine the Ronne Ice Shelf, reducing its buttressing effect and allowing the ice streams to accelerate. The timeline for this process is uncertain. Some models suggest that the Ronne Ice Shelf could remain stable for centuries under moderate warming scenarios, providing continued protection for the ice streams behind it. Other models predict more rapid deterioration, with significant weakening possible within decades under high emissions scenarios. The discrepancy between model predictions underscores the urgent need for observational data, both from the modern ice sheet and from the geological record, to constrain the models and improve their predictive capability. The Salsa sediment record contributes to this effort by providing a direct constraint on past ice sheet behavior that models must reproduce. As more data from the Lake Mercer cores becomes available, including improved age estimates from cosmogenic nuclide and luminescence dating, the constraints will tighten, and the models that best reproduce the observed history will gain credibility as tools for predicting the future. The exploration of the Lake Mercer region and other parts of the West Antarctic ice sheet is therefore not an academic exercise. It is a critical component of the scientific effort to predict future sea level rise and prepare for its consequences. The sediments at the bottom of a subglacial lake beneath a kilometre of ice, recovered at enormous expense by a team of scientists working in one of the most extreme environments on Earth, contain information that is directly relevant to the lives and futures of billions of people living in coastal regions around the world. The connection between the subglacial sediments and global sea level rise is mediated by the ice sheet models, which translate paleoclimate evidence into projections of future change. Improving these models by testing them against the Lake Mercer data and other observational constraints is one of the most important tasks in contemporary climate science, and the explorations and attempts that have brought us to this point. From the first Soviet radar surveys of the 1960s through the Wissard success and the Salsa discovery, represent a sustained investment of human ingenuity, resources, and determination that is now beginning to pay dividends in the form of actionable knowledge about the future of the ice. The pattern is clear. Each mission reveals more than the previous one. Each set of data raises questions that the previous dataset could not address, and each new insight into the behavior of the ice sheet strengthens the scientific foundation on which policy decisions about climate change, sea level rise, and coastal planning must be based. The explorations are not complete. They may never be complete, but they are making progress slowly, painstakingly, one borehole at a time, one sediment core at a time, one analytical result at a time, toward the understanding that the fate of the frozen continent demands. The John Mercer legacy deserves a moment of reflection in the context of the exploration that bears his name. Mercer was among the first scientists to warn publicly about the vulnerability of the West Antarctic ice sheet to collapse under global warming. His 1978 paper, published in the journal Nature, argued that the burning of fossil fuels could raise atmospheric carbon dioxide levels sufficiently to warm the Southern Ocean, melt the ice shelves buttressing the West Antarctic ice sheet, and trigger a disintegration that could raise global sea levels by several meters within a few centuries. At the time, Mercer's warning was received with skepticism by much of the scientific community. The mechanisms he described were poorly understood. The climate models of the era were too crude to test his hypothesis rigorously, and the observational evidence was insufficient to confirm or refute his predictions. His paper was noted, but not widely embraced. The scientific community preferred to wait for more data before endorsing what seemed like an alarmist scenario. 4 decades later, the data has arrived. The satellite observations, the ice sheet models, the ocean temperature measurements, and now the sediment record from the subglacial lake that bears Mercer's name all point in the same direction. The West Antarctic ice sheet is vulnerable. It has retreated before, and the conditions under which it retreated in the past are being recreated by human activities at a rate that exceeds anything in the recent geological record. Mercer did not live to see the vindication of his hypothesis. He died in 1987, more than 3 decades before the Salsa team drilled through the ice above the lake, named in his honor, and recovered the sediments that provided the most direct evidence yet for the dynamism he had predicted. But his intellectual legacy lives on in the research program that the Salsa discovery has stimulated, a program that is driven by the same concern that animated Mercer's original paper, the fear that human activities are pushing the Earth's climate system toward a threshold beyond which the great ice sheets of the polar regions begin to disintegrate. The irony of the Lake Mercer discovery is that the sediments contain both a warning and a measure of hope. The warning is that the ice has retreated before, that the landscape beneath the ice sheet is not permanently frozen but intermittently exposed, and that the forces driving the current retreat, warming temperatures and warming oceans, are the same forces that drove past retreats. The measure of hope is that after each past retreat, the ice returned, the ice sheet reformed, the subglacial environment was resealed. The cycle of retreat and advance continued without the permanent loss of the ice sheet. But this hope must be qualified by the recognition that the current warming is different from past warming events in both its speed and its cause. Past interglacial warm periods were driven by slow changes in Earth's orbital parameters, changes that unfolded over thousands of years and gave the climate system time to adjust gradually. The current warming is driven by the rapid release of greenhouse gases from the burning of fossil fuels, a process that is occurring over decades, not millennia. The speed of the forcing may overwhelm the ice sheet's capacity to respond gradually, potentially triggering rapid, irreversible retreat that no natural interglacial has ever produced. The Lake Mercer sediments cannot tell us whether the current warming will trigger such a collapse. That question can only be answered by the models, constrained by the observations, and verified by time. But the sediments can tell us that the ice sheet is capable of retreating from this location under conditions that are broadly comparable to those that human activities are creating. And that knowledge, combined with the satellite observations of accelerative ice loss and the modeling results that project continued deterioration, constitutes the most powerful evidence available for the urgency of reducing greenhouse gas emissions and preparing for the consequences of the warming that has already occurred. The explorations at Lake Mercer have brought us to the boundary between what we know and what we need to know. On one side of that boundary lies the evidence already in hand: the living microbes, the fossil remains, the sediment chemistry, the dating results, the satellite observations, the model simulations. On the other side lies the understanding we have not yet achieved: the precise timing of past ice-free conditions, the mechanisms that drove past retreats, the thresholds that control the ice sheet's sensitivity to warming, and the timescales over which future retreat might unfold. Crossing that boundary will require more missions, more data, more analysis, and more time. But the direction of travel is clear, and the urgency is undeniable. The organisms in the Lake Mercer sediments, the crustaceans and diatoms and tardigrades that lived at this site when the ice was absent, are not merely scientific specimens. They are witnesses to a past that the current trajectory of human emissions threatens to recreate, and their testimony, preserved in a few centimeters of mud beneath a kilometer of ice, is among the most important evidence that science has ever recovered about the future of the planet. The deeper truth of Lake Mercer is not about crustaceans or diatoms. It is not about drilling technology or contamination control. It is not even about the stability of the West Antarctic ice sheet, though that issue is among the most consequential facing human civilization. The deeper truth is about the relationship between life and ice, between biology and climate, between the living and the frozen, and about what that relationship reveals about the nature of our planet and the fragility of the conditions that sustain us. Consider what the Lake Mercer sediments tell us about the resilience of life in the face of catastrophic environmental change. The organisms whose remains were found in the sediments, the crustaceans, the diatoms, the tardigrades, the mosses, lived during a time when the climate at this location was warm enough to support open water and terrestrial vegetation. Then the ice came. The temperature dropped. The sunlight was blocked. The water pressure increased 100-fold. Every condition that had sustained these organisms was eliminated in a geological instant, replaced by a dark, frozen, pressurized environment that was hostile to every form of surface life. The photosynthetic organisms died, the grazing animals died, the terrestrial plants died, but life itself did not die, it was replaced. The chemolitho-autotrophic microbes, organisms capable of deriving energy from chemical reactions rather than sunlight, colonized the new subglacial environment and established a functioning ecosystem in conditions that would have killed every organism that preceded them. The biological world beneath the ice was not a continuation of the biological world that had existed in the open. It was a replacement, a fundamentally different mode of life occupying the same physical space under radically different conditions. And when the ice retreated, as the sediments suggest it did at least once, and possibly multiple times, the process reversed. The chemolitho-autotrophic community was marginalized or eliminated, and photosynthetic organisms, carried in by wind, water, or animal vectors, recolonized the newly exposed landscape. Diatoms bloomed in the sunlit water, crustaceans grazed on the algae, mosses colonized the exposed soil. A new ecosystem, fundamentally different from the one that had existed beneath the ice, established itself in what had been, just years or decades earlier, a dark, frozen, lifeless world. Let that sink in. The same physical location has hosted two completely different modes of life, one driven by sunlight and one driven by chemical energy, each replacing the other as the ice advanced and retreated. The location has been a stage on which two fundamentally different biological dramas have been performed, each one erasing the cast of the previous performance and installing a new one. The crustaceans and diatoms in the sediments are the fossilized cast of one performance. The chemolitho-autotrophic microbes in the current lake water are the living cast of the performance now underway, and beneath them, in layers not yet reached by the sulfur drilling, there may be evidence of earlier performances, earlier cycles of ice and openness, earlier alternations between sunlit and dark ecosystems. This cyclic replacement of one mode of life by another is a phenomenon of extraordinary scientific and philosophical significance. It demonstrates that life is not a single, unified process but a spectrum of strategies, each adapted to a different set of environmental conditions. The photosynthetic mode, which dominates Earth's surface and drives the global carbon cycle, is not the only way to be alive. The chemolitho-autotrophic mode, which operates in the dark, beneath ice and rock, sustained by the chemical energy of the Earth's interior, is equally valid, equally functional, and potentially far more widespread in the cosmos than the photosynthetic mode. This is not science fiction. This is empirical biology, documented in the sediment record of a glacial lake and confirmed by the analysis of the living community that currently inhabits. The Lake Mercer discovery provides the clearest evidence yet that life on Earth operates in two fundamentally different modes, one dependent on the sun and one independent of it, and that these two modes can alternate at a single location as conditions change. The deeper truth extends to the question of time and permanence. The ice sheet that currently covers the Lake Mercer site is massive, dense, and seemingly permanent. From a human perspective, it is a fixture of the landscape, as immovable and enduring as the mountains. But here's the problem. The sediments tell a different story. They tell a story of transience, of a landscape that oscillates between frozen and unfrozen states, of ice that comes and goes on timescales that are long by human standards but brief by geological ones. The ice is not permanent. It is a phase, a temporary condition maintained by a specific set of climate parameters that have not always prevailed and will not always prevail. The transience of the ice raises questions that are not merely scientific but existential. The human species has existed for roughly 300,000 years. Agriculture has existed for roughly 10,000. Industrial civilization has existed for roughly 250. In the span of time during which humans have been building the coastal infrastructure that is now threatened by sea level rise, the West Antarctic ice sheet has appeared to be a permanent, unchanging feature of the planet. But the geological record, preserved in the Lake Mercer sediments, reveals that this appearance of permanence is an illusion created by the brevity of human observation. The ice sheet has been advancing and retreating for millions of years, driven by forces that operate on timescales far longer than human history. The current configuration of the ice sheet, with ice covering the Lake Mercer site and the broader West Antarctic interior, is a snapshot in a long-running geological process, not an endpoint. The ice will change. The coastlines will change. The geography of the planet will change. The only question is when and how fast, and whether human activities are accelerating the change beyond what natural forcing alone would produce. The answer to the last question, based on the evidence available, appears to be yes. The current rate of warming, driven by anthropogenic greenhouse gas emissions, exceeds anything in the recent geological record. The atmospheric concentration of carbon dioxide, now exceeding 420 parts per million, is higher than at any time in at least 800,000 years, based on ice core records. The warming that this concentration implies, amplified by feedback mechanisms, including ice albedo feedback, water vapor feedback, and the release of methane from thawing permafrost and subglacial reserves, is pushing the climate system toward conditions that have not existed since long before the current glacial period began. The Lake Mercer sediments provide a window into what those conditions looked like at one location beneath the ice. The picture is both beautiful and alarming. Beautiful because it reveals a world of open water and biological diversity, a landscape alive with photosynthetic algae and microscopic animals and plants, thriving in conditions that supported the full range of biological activity. Alarming because it reveals that this world existed at a location that is now buried beneath a kilometre of ice, and that the creation of this world required the removal of the ice, which in turn required the kind of warming that is now underway. The deeper truth connects the Lake Mercer discovery to every aspect of the human relationship with the planet's climate system. The ice sheets are not decorative features of the polar regions. They are functional components of the Earth system, reflecting sunlight back into space, regulating ocean circulation, moderating global temperatures, and storing enormous volumes of water that would otherwise be in the ocean, raising sea levels and flooding coastal regions. The removal of the ice sheets, or even a significant reduction in their extent, would alter the Earth system in ways that would affect every human being on the planet. The Lake Mercer sediments remind us that this alteration has happened before, naturally, in response to climate forcing that was modest by comparison to what human activities are now producing. The organisms in the sediments lived in a world where the ice was gone. That world was not catastrophic for the organisms that inhabited it. They were adapted to it. They thrived in it. But for human civilization, built on the assumption of stable coastlines, stable ice sheets, and a stable climate, the return of that world would be transformative in ways that are difficult to overstate. The deeper truth is therefore a truth about vulnerability. The vulnerability of the ice to warming, the vulnerability of the coastlines to sea level rise, the vulnerability of human civilization to changes in the physical landscape that it has taken for granted, and the vulnerability of the subglacial biosphere itself, a hidden biological world that has been functioning in the dark for millennia and that may be altered or destroyed by the same warming that threatens the ice above it. This recognition of the ice sheet's transience has implications that extend beyond climate science to the realm of philosophy and ethics. If the ice is transient, then the organisms that live beneath it are also transient, their existence contingent on the maintenance of conditions that can change. And the organisms that live in the open, including human beings, are equally contingent, their existence sustained by a different set of conditions that are themselves subject to change. The deeper truth is that nothing about the current state of the Earth's surface is permanent. The ice sheets, the coastlines, the ecosystems, the climate zones, all of these are features of a temporary configuration, a snapshot of a planet in motion. The Lake Mercer sediments remind us that this snapshot has been different in the past, and will be different in the future, and that the changes from one snapshot to the next can be dramatic, transforming an ice-free landscape into a frozen one, or a frozen landscape into an open one within the span of centuries to millennia. Let that sink in. The landscape that we see today, the ice sheet that we depend upon to remain in place, the coastlines that we have built our cities along, all of these are temporary. They are features of the current climate, and when the climate changes, as it has changed in the past and is changing now, these features will change with it. The organisms in the Lake Mercer sediments knew a different world, a world without ice, a world of open water and sunlight at a location that is now buried beneath more than a kilometre of frozen darkness. That world existed within the recent geological past, and the conditions that could recreate it are being generated by human activities right now. The deeper truth of Lake Mercer is therefore a truth about the relationship between human civilization and the physical systems that sustain it. We have built our cities, our economies, our transportation networks, and our food systems on the assumption that certain features of the physical landscape are permanent, that the coastlines will remain where they are, that the ice sheets will remain in place, that the climate will remain within the range of conditions to which our infrastructure and institutions are adapted. The Lake Mercer sediments challenge this assumption with the authority of the geological record, demonstrating that the features we take for granted are neither permanent nor stable, and that the forces capable of transforming them are already at work. The connection between the ancient organisms in the sediments and the modern crisis of climate change is not metaphorical, it is causal. The same greenhouse gas, carbon dioxide, that is being released by the burning of our fossil fuels is the same gas that, at higher concentrations in the atmosphere, creates the warming that can destabilize ice sheets. The relationship between atmospheric carbon dioxide concentration and Antarctic ice sheet behavior is one of the best documented relationships in all of Earth science, supported by ice core records, marine sediment records, and the fundamental physics of the greenhouse effect. The Lake Mercer sediments add another layer to this body of evidence, showing that the consequences of elevated carbon dioxide levels include the retreat of ice from regions that are currently frozen. The ethical dimensions of this knowledge are significant. We know from the geological record that the ice sheet is capable of retreating from the Lake Mercer region under conditions of modest warming. We know from satellite observations that the ice sheet is currently losing mass at an accelerating rate. We know from climate models that continued greenhouse gas emissions will produce warming sufficient to destabilize significant portions of the West Antarctic ice sheet. And we know from the sediments of Lake Mercer what the landscape looks like when the ice goes away. With this knowledge comes responsibility. The decisions that human civilization makes about greenhouse gas emissions in the coming decade will determine whether the ice sheet at the Lake Mercer site remains in place or retreats. If the ice retreats, the consequences will include sea level rise measured in meters, affecting hundreds of millions of people around the world. The organisms in the Lake Mercer sediments, the crustaceans and diatoms and tardigrades that once lived in open water at this location, are not merely scientific specimens. They are a message from the geological past, a warning that the ice can go away, written in the language of tiny shells and algal remains, sealed beneath a kilometre of the very ice that their existence calls into question. The deeper truth is that the past is not past, it is prologue. The conditions that created the world recorded in the Lake Mercer sediments are being recreated by human activities, not over millennia, but over decades. Whether the outcome will be the same, whether the ice will retreat again and the open water will return, depends on choices that are being made right now, in the halls of government, in the boardrooms of corporations, and in the daily decisions of billions of individuals around the world. The crustaceans in the sediments had no say in the climate that created their world. We do, and the question is whether we will exercise that agency wisely enough to preserve the frozen world that currently exists above their remains. The implications of the Lake Mercer discovery extend through multiple layers of significance, from the immediate scientific findings to the broadest questions about humanity's relationship with the natural world. Each layer carries its own weight, and together they form a structure of meaning that reaches from the sediments of a subglacial lake to the future of human civilization. The first layer is scientific. The Lake Mercer sediments have demonstrated that the West Antarctic ice sheet is more dynamic than many previous assessments had assumed, capable of retreating from regions that are currently covered by more than a kilometre of ice. This finding constrains ice sheet models, informs projections of future sea level rise, and motivates additional research into the mechanisms and timescales of ice sheet retreat. The scientific implications are direct, measurable, and actionable, providing data that can be incorporated into the models and projections that guide policy decisions about climate change adaptation and mitigation. The second layer is planetary. The coexistence of living microbes and ancient biological remains in the Lake Mercer environment demonstrates that ice-sealed aquatic environments can preserve a rich biological record spanning multiple environmental regimes. This finding is directly relevant to the design of future missions to the icy moons of the outer solar system, where similar environments may exist, and where similar preservation of biological evidence may have occurred. The Lake Mercer discovery strengthens the scientific case for missions to Europa and Enceladus, providing a terrestrial proof of concept for the detection of biosignatures in ice-sealed aquatic environments. The third layer is ethical. The knowledge that the West Antarctic ice sheet has retreated in the past, and that the conditions for retreat are being recreated by human activities, places a moral burden on the current generation. We are not merely observing a natural phenomenon. We are contributing to the forces that may cause it to recur. The greenhouse gas emissions produced by our energy systems, our transportation, our agriculture, and our industry are altering the climate in ways that threaten the stability of the ice sheets, the persistence of the coastlines, and the well-being of hundreds of millions of people who live in low-lying regions around the world. This ethical dimension is sharpened by the Lake Mercer discovery because the sediments provide specific, tangible evidence of what happens when the ice retreats. The crustacean shells and diatom frustules in the sediments are not abstractions. They are the physical remains of organisms that lived in a world without ice at this location. They give concrete form to the consequences of ice sheet retreat, making the abstract concept of sea level rise tangible and immediate. The fourth layer is temporal. The Lake Mercer sediments connect past, present, and future in a way that few other scientific findings can match. The past is recorded in the biological remains and chemical signatures of ice-free conditions. The present is embodied in the living microbial community and the accelerating loss of ice observed by satellites. And the future is implied by the convergence of these two sets of evidence, which together suggest that the trajectory of change is toward further retreat, further ice loss, and further sea level rise. The temporal perspective is humbling. The organisms in the sediments lived during a period that is beyond the reach of human memory, historical record, or even oral tradition. They inhabited a world that no human being has ever seen, a world that exists only in the geological record and in the inferences that scientists draw from tiny fragments of shell and organic matter preserved in subglacial mud. And yet the forces that created that world, the warming of the climate and the retreat of the ice, are the same forces that are operating now, driven not by orbital variations, but by the choices of a civilization that has the knowledge to understand the consequences, but has not yet demonstrated the will to change course. The reflection that Lake Mercer demands is a reflection on the adequacy of our response to what we know. We know that the ice sheet is losing mass. We know that the rate of loss is accelerating. We know that the sediment record shows that the ice has retreated from this location before. We know that the warming that is driving the current changes is caused by human activities. And we know that the consequences of continued inaction include sea level rise that would reshape the geography of the planet and displace hundreds of millions of people. What we do not know is whether this knowledge will be sufficient to motivate the changes in behavior, policy, and economic structure that are needed to reduce greenhouse gas emissions quickly enough to prevent the worst outcomes. The history of human responses to environmental warnings is mixed, marked by both remarkable successes and spectacular failures. The Lake Mercer discovery adds another voice to the growing chorus of evidence calling for action. But whether that voice will be heard above the noise of competing interests, short-term thinking, and institutional inertia remains to be seen. The scientific community has done its part. The SALSA team travelled to one of the most hostile environments on Earth, drilled through a kilometre of ice, recovered samples that no one had thought possible a generation ago, and produced findings that have reshaped our understanding of ice sheet dynamics and climate history. The samples have been analysed with the most sophisticated instruments available. The results have been published in the peer-reviewed literature. The implications have been communicated to the public and to policymakers. The reflection that these findings demand is not merely intellectual, it is personal. Every person living on a coastline, in a river delta, on a low-lying island, or in a region where the economy depends on stable sea levels, has a stake in the questions that the Lake Mercer sediments have raised. The crustacean shells and diatom frustules in the sediments are not abstract scientific data. They are evidence that the physical foundation of coastal civilization, the assumption that the ice sheets will remain in place and the coastlines will remain where they are, is not as solid as we have assumed. The psychological challenge of absorbing this information is real. Human beings are not well equipped to respond to threats that unfold over decades and centuries rather than hours and days. The ice sheet does not retreat like a wildfire or a flood, visible and immediate and demanding instant response. It retreats slowly, imperceptibly, a few meters of grounding line per year, a fraction of a millimeter of sea level rise per month. The cumulative effect over decades and centuries is enormous, but the daily change is too small to notice, too gradual to trigger the alarm responses that evolution has equipped us with. The Lake Mercer sediments offer a corrective to this psychological limitation. They compress the consequences of ice sheet retreat into a single, tangible set of evidence. Organisms that lived in a world without ice, at a location that is now covered by a kilometre of frozen water. The compression of a geological process into a handful of sediment cores makes the abstract concrete, the gradual sudden, the distant immediate. The crustaceans in the sediment are a message from a world that the retreat of the ice would recreate. And the retreat of the ice, driven by the warming we are causing, is already underway. The next steps are not scientific. They are political, economic, and social. The knowledge generated by the Lake Mercer discovery must be translated into policy decisions that reflect the urgency of the situation. The sea level rise projections informed by the SALSA data must be incorporated into infrastructure planning, building codes, and land use regulations. And the broader lesson of the discovery, that the great ice sheets of the polar regions are not permanent fixtures, but dynamic features capable of rapid change, must be absorbed into the public consciousness in a way that motivates individual and collective action. The organisms in the Lake Mercer sediments cannot tell us what to do. They can only tell us what has happened. The crustaceans and diatoms and tardigrades lived in a world without ice, and then the ice came, and they died, and their remains were buried in sediment that was sealed beneath a kilometre of frozen water for thousands of years. That is their story. Our story, the story of what we do with the knowledge their remains have provided, has not yet been written, but the time for writing it is growing short, and the sediments beneath the ice, patient as they are, will not wait forever for us to learn their lesson. Beneath the Mercer ice stream, in a thin lens of water pressed between ice above and rock below, in a darkness that has not been broken since, long before any living person was born, the lake persists. Its water circulates slowly, moved by pressure gradients and geothermal heat. Its microbes metabolize, consuming the chemical energy of iron and sulfur compounds in the sediments, fixing carbon dioxide into organic molecules, producing methane and carbon dioxide as metabolic waste. And in the mud at the bottom, undisturbed by wind or wave, the remains of creatures from another time lie where they settled, shell beside shell, frustule beside frustule, silent witnesses to a world that no longer exists. The crustaceans in the sediment do not know that they are evidence. They do not know that their tiny shells, barely visible to the naked eye, carry the weight of one of the most consequential findings in the history of Antarctic science. They do not know that their presence beneath a kilometre of ice has challenged the assumptions of glaciologists, alarmed the modelers of sea level rise, and provided ammunition for those who argue that the West Antarctic ice sheet is more vulnerable to collapse than the world has been prepared to accept. They are simply there, preserved in mud, remnants of a life lived in sunlight at a place that is now buried in ice. The tardigrades in the sediment do not know that their fragments have inspired wonder in the researchers who found them. These microscopic animals, famous for their ability to survive extreme conditions in a dormant state, were found not as living survivors of the glaciation but as dead remains, their bodies preserved in the sediment like tiny fossils in a geological archive. They had lived during the ice-free period, swimming or crawling through the shallow water of a surface environment that has since been sealed beneath a kilometre of frozen darkness. Their survival skills, legendary in the popular imagination, were insufficient to save them from the advancing ice. The ice does not negotiate, it does not make exceptions. It comes, and what it covers it buries. The diatoms in the sediment do not know that their frustules, the tiny silica shells that each diatom constructs during its lifetime, are among the most important pieces of evidence ever recovered from beneath the Antarctic ice sheet. These photosynthetic algae required sunlight to survive. Their presence in the sediments is therefore proof, as direct and as unambiguous as the geological record can provide, that sunlight once reached this location, that the ice was absent, that the sky was visible, that the conditions that we see today, the kilometre of ice, the total darkness, the crushing pressure, are not permanent features of this landscape but temporary ones, maintained by a climate regime that has not always prevailed and may not always prevail. The message of the Lake Mercer sediments is simple and profound. The ice has not always been here, the ice may not always be here, and the conditions that determine whether the ice stays or goes are within the influence, if not the control, of human civilization. This is the message that the crustaceans and diatoms and tardigrades have delivered from their resting place beneath the ice. It is a message written in the language of biology, in the grammar of tiny shells and organic fragments, in the syntax of sediment layers alternating between glacial and non-glacial conditions. It is a message that has travelled from a world we have never seen to a world we are at risk of creating. And it is a message that, if we are wise enough to read it, may yet change the trajectory of our species and the future of the ice that covers the bottom of the world. The lake is dark, the water is cold, the pressure is immense, but the sediments remember. They remember a time of light, of warmth, of open water, of life in its sunlit, photosynthetic, familiar form. They remember a world without ice, a world that existed at this very spot, in this very basin, beneath this very sky, during a chapter of Earth's history that has closed but may yet reopen. The closing thought is not about endings, it is about beginnings, because the Lake Mercer discovery is not the conclusion of a scientific story. It is the opening of a new chapter, a chapter that will be written by the choices we make about the climate, by the investments we make in understanding the ice, and by the seriousness with which we take the warnings that the frozen continent has been sending us from beneath its eternal, implacable, deceptively permanent ice. The ice is patient. The sediments are patient. The crustacean shells and diatom frustules are patient. They have waited for thousands of years to deliver their message. They will wait a while longer. But the planet they lived on will not wait forever. And the ice above them, the ice that sealed their world and preserved their remains, is thinning. It is warming. It is, in ways that are measurable and documented, beginning to lose its grip on the continent beneath. What happens next is not written in the sediments. It is written in the choices we make today, tomorrow, and in the decades to come. The ancient life beneath the ice has spoken. The question is whether we will listen. We stand at a peculiar moment in the history of our species. We have, for the first time, the ability to see what lies beneath the ice, to recover the records that the subglacial world has preserved, to read the geological diaries that have been sealed in mud for millennia. We have sent teams to the most remote places on Earth, developed technologies capable of penetrating kilometers of ice, without contaminating what lies beneath, and produced scientific findings of a quality and significance that would have astonished the first Antarctic explorers. We have done something extraordinary. We have heard the voices of organisms that lived in a world we have never seen, speaking to us across an abyss of time through the medium of tiny shells and organic fragments. And those voices are saying something urgent. They are saying that the ice is not forever, that the world beneath it has been open before, that the conditions for openness are being created again, this time by us, and that the consequences of ignoring their message, of continuing to pour greenhouse gases into the atmosphere, as if the great ice sheets were immune to the warming we are causing, will be measured not in abstract statistics, but in the flooding of cities, the displacement of populations, and the transformation of coastlines that billions of people call home. The sediments are patient. But patience is not the same as indifference. The organisms in the mud lived their lives, died their deaths, and left their remains for us to find, not as a deliberate message but as an accidental archive, a record of conditions that once existed and may exist again. Whether that record becomes a warning heeded or a warning ignored depends entirely on us. The ice above is thinning, the ocean beneath is warming, and the ancient life in the sediments, silent and still, waits for the world to catch up to what the science has already shown.